Okay, well, hello everyone. Today I'm going to be presenting this paper called Recent Advances in Alzheimer's Disease, Mechanisms, Clinical Trials, and New Drug Development Strategies. It's a very long review paper. It's like 25 pages. It has over 250 cited references that they include in the paper. And I made a choice when I picked this paper, which was to read and learn about a biology-based paper as someone who doesn't understand biology at all. So when I was making this PowerPoint, I ran into a lot of challenges. So I decided to only present the mechanisms part of the paper to make it a little bit easier for myself. And I will go ahead and give that note now that I am not a biology person. I tried to learn most of this week. So hopefully I've had it reviewed by someone who is or has a biology background and they said everything looked good. But if you guys know anything that I may be saying wrong or want to jump in with the notes, please do. So that's my disclaimer at the very beginning.
Vinny Jordan.
Yes, Vinny Jordan. Actually, one of the mechanisms I'm going to be reviewing is one that he's presented on a bunch of times. I think he would have a good bit to say about it. So I'll start with a really quick introduction that they include in the paper, which is that Alzheimer's disease is the predominant form of dementia. In the US, approximately 10% of adults age 65 or above suffer from Alzheimer's disease. And by 2030, the estimated number of people living with dementia globally is going to exceed 78 million. There's also a big global burden that's associated with the medical care, the social services, and the informal caregiving for those with dementia, and that is above 28 trillion US dollars. So it's a large amount. And Alzheimer itself is characterized by memory loss, cognitive decline, behavioral disturbances, and ultimately a loss of independence. Due to its clinical and biological complexities, Alzheimer's presents major challenges for therapeutic development, and there is still lots of work being done, and nobody really has the perfect therapy or cure for it yet. Alzheimer's disease can be characterized into two main types. There is the familial Alzheimer's disease, which is about 1 to 5% of cases. It's characterized by specific autosomal dominant genetic mutations, and these three, one of which we're going to be precursor protein. And it typically manifests between 30 to 65 years of age, and it progresses really quick. The second type is the sporadic or late onset Alzheimer's, which is over 95% of cases. It's influenced by a combination of genetic risks, environmental factors, and various comorbidities, like maybe diabetes or autoimmune disorder or something like that. And it typically manifests after the age of 65. This is kind of a progression chart of Alzheimer's. It starts in the preclinical stage where pathological changes in the brain begin. It's sometimes decades before clinical symptoms even show. There is a stage between preclinical and mild, some include it as an actual stage, some just mention it and don't include it, which is the mild cognitive impairment stage. This is where you see the first notable signs of decline in cognitive skills, but daily function is mostly the same. It doesn't yet meet the criteria for dementia, so some resources that I visited did not include it in this progression. But it's a really critical period for early diagnosis and potential therapies if we know that it's going to become Alzheimer's later on or develop into Alzheimer's. There is the mild stage where symptoms include mild forgetfulness, problems with concentration maybe. It progresses to the moderate stage that has more severe memory loss. Here you start seeing neuropsychiatric symptoms such as hallucinations and intensified emotional and behavioral issues. And then finally, it progresses to the severe stage where you see a lot of loss of language skills, inability to perform daily activities, and complete reliance on caregivers. This is a helpful chart that the paper provides, and it summarizes the 9 mechanisms, or it calls it hypotheses of Alzheimer's disease that it's going to go through. Here it couples 2 of them together, so it shows 8, but it discusses them in 9, so I'll discuss them in 9. And An important thing to notice that will become clear as I describe these is that these hypotheses are by no means separate, or that they don't like work independently of each other. They all work together, they influence each other, they interact with each other to help develop Alzheimer's. But it's helpful to look at them separately because the therapies and the treatments are different for each one. And so maybe if you can focus on each one separately, you can come up with a cure for all of it, or treatment for all of it. So we're going to start with the cholinergic hypothesis. The cholinergic hypothesis is one of the more well-established theories of Alzheimer's disease and kind of the earliest to describe the pathogenesis of Alzheimer's. The theory is centered around cholinergic neurons, which are neurons that synthesize and release the neurotransmitter acetylcholine. Now, acetylcholine plays a very important role in physical functions like motor control and cognitive functions like attention, memory, learning, arousal, and sleep-wake regulation, which are all of those things that are impacted by Alzheimer's. And just by those names, I think you can tell that it's a big deal. Cholinergic neurons synthesize acetylcholine using the enzyme choline acetyltransferase, called CHAT for short. It has been observed that severe damage of cholinergic neurons at the nucleus basalis of Maynard, which is that little part pointed on at the figure at the top, leads to a marked decrease in CHAT activity in the cerebral cortex and the hippocampus, which are regions associated with learning and memory. Acetylcholine is broken down by the enzyme acetylcholinesterase, AIC, I think it's pronounced. So the main therapies that are being developed in this framework are AIC inhibitors, which block the enzyme that breaks down acetylcholine so that there is a greater level of acetylcholine presence in the brain, as well as a longer activation level of those acetylcholine in the brain. These drugs include, I'm going to butcher these names, donepezil, rivastigmine, and galantamine, and they're kind of the mainstay of Alzheimer's treatment so far. Maybe you've heard of this before, I'm not sure. However, some of these drugs have a limited efficacy or really strong side effects, and there are also certain non-cholinergic groups of Alzheimer's for which this treatment doesn't work, or any therapies of this hypothesis don't work. There is still work being done with optimizing the doses, the dosage forms, the routes of administration. For example, if I remember correctly, the paper mentioned that in 2022, the FDA approved a weekly patch version of donepezil, which showed to have the same effect as the oral pill, but without any of the adverse gastrointestinal side effects. So work is still being done in this hypothesis, and it's kind of most of the treatments you see are from this framework. The next one, which I think is the one that most people are familiar with, is the amyloid hypothesis. So let's start with the fact that the accumulation of amyloid beta is a hallmark pathological feature in AD. How does that even become to creation? There are two enzymes, beta secretase and gamma secretase, that cleave the amyloid precursor protein, called APP, into various lengths of amyloid beta fragments. There are two very common fragments that are produced by this process. There is AB40 and AB42. AB40 is generally more prevalent, but AB42 is less soluble and more prone to aggregation. So it's not efficiently broken down and accumulates to form the core of senile plaques over time as people age. Now, it's important to note that this process occurs in everyone as they age to some extent, but it is significantly accelerated in people with Alzheimer's disease. Researchers have identified a set of mutations and genetic risk factors that can cause this acceleration. And one of the most important genetic risk factors for specifically sporadic or late onset Alzheimer's, which if you remember is at 95% of cases, is apolipoprotein E. ApoE, and particularly its epsilon-4 allele. The paper states that just carrying one ApoE, epsilon-4 allele increases the risk of Alzheimer's disease by 2 to 3-fold, and carrying two of those exposes the risk or increases the risk by 12-fold. So huge increase that comes along with that. And on the figure here, you can see the areas in yellow, which are areas more atrophied in all patients with Alzheimer's disease compared to controls. And there are also red regions which show areas or regions that are more atrophied in those with epsilon-4 carriers compared to those who don't have it. So you can see it captures quite a few more regions, and so it's a really important factor that researchers have found. There is a hypothesis, a second hypothesis at the core of this one, which is called the amyloid cascade hypothesis. And it has stated that the accumulation of amyloid beta protein in the brain is the primary driver or the primary cause of Alzheimer's. Now, this has been a highly contentious and controversial hypothesis, but it has led to the development of several therapies, including inhibitors for the beta secretase and gamma secretase, amyloid beta vaccines, and more. And recently, some antibodies like aducanumab, lecanemab, and donanemab have shown promise in supporting amyloid beta's role in Alzheimer's disease pathogenesis by targeting and reducing amyloid plaques. And when the paper was published, at the time this paper was published, it mentioned that donanemab had completed phase 3 trials and was in the process of market authorization. And I thought it was pretty cool that just a few days ago, Alex sent this paper to one of the Slack channels that specifically talked about its use and recommendations and how it's been approved as a new cool therapy for this specific paper. If anyone has any questions. The next hypothesis that we're going to look at, which I think people are also commonly familiar with, is the tau protein hypothesis. And so let's start by looking at what tau even is. Tau is a microtubule-associated protein that helps stabilize microtubules in neurons. So what are microtubules? You can see a visualization of one in the top right there. They're essentially hollow cylindrical structures that provide support and shape to various cells. So they can help facilitate intracellular transport, other cell functions like cell division, movement of food. So they support a lot of really important functions in cells, and they're held up or stabilized by the tau protein. I will give, once again, an extra note here that they are very important structures for various other functions, so not just in this specific case. Tau proteins have multiple phosphorylation sites that are regulated by kinases and phosphatases to support normal neuronal functions. Phosphorylation itself refers to the process of simply adding a phosphate group to a protein or another molecule. And phosphates act like a molecular switch for different functions. So they're like turning functions on and off, essentially. Or they modify how proteins interact with other molecules. Okay. Now, in Alzheimer's disease, there can be disruptions of phosphatase activity, which leads to hyperphosphorylation of tau proteins. When the tau proteins become hyperphosphorylized, first, they detach from microtubules. And when they detach from microtubules, they're floating around, they aggregate with paired helical filaments and other tau proteins, and they form neurofibrillary tangles, called NFTs for short, inside neurons. These tangles act similarly, they're not plaques, but they act similarly to plaques in that they act like a physical barrier and prevent normal neuronal function, stop things from passing through, or essentially a big roadblock that eventually leads to cell death. In cell death, I think I didn't explicitly say it, or maybe I did, I don't remember, but cell death is kind of the big thing that's happening in Alzheimer's. Cells are dying, therefore cognitive or neurons specifically are dying in the brain, so different functions are disrupted by that process. So anything that leads to cell death is what they're trying to stop. So not only that, but when the tau proteins detach, the microtubules no longer have anything to stabilize them. So they collapse and they can lead to impaired neuronal transport. Things don't move smoothly anymore. Things can be dysfunctional. Other processes are dysfunctional, and that also leads to cell death. Here I've included a note that says the significance of tau and AD pathogenesis lies in that there's a really strong correlation between the tau accumulation or the amount of tau presence and the severity of cognitive symptoms. The paper mentions that initial investigations into drugs based on this hypothesis have not yielded promising results, but that there are numerous treatments that are still being actively developed. I've included two images on the screen. The one on the bottom left shows some amyloid plaques and neurofibrillary tangles in an Alzheimer's brain compared to a normal brain. And the picture on the right is a figure with amyloid beta plaques on the top, so those are those round structures, and showing them as one potential cause for tau hyperphosphorylation and the process that occurs as a result of hyperphosphorylation, the neurofibrillary tangles, and then synaptic dysfunction neurons follow. I showed this to someone to ask their feedback on it, and they said that the figure is stupid for various reasons, and that it doesn't represent things accurately, but I still think it's helpful because it starts showing how all of these theories are interconnected and there's an interplay between them. So there is some sort of relationship between amyloid beta plaques and the tau protein, but it's not as cause and effect as this makes sense. Moving on to the next hypothesis, we have the neuroinflammation hypothesis. Neuroinflammation is defined as a chronic inflammatory response in the central nervous system that fails to resolve on its own. We all have glial cells in our body, and glial cells are responsible for providing chemical and physical support to neurons to help them maintain their environment. There are 5 types of glial cells, and one of those is microglia. Microglia are the immune cells of the central nervous system. They see bad stuff, they attack bad. So inflammation in its standard amounts is normal, a pretty healthy mechanism. But hyperactivation of microglia can lead to excessive inflammation and contribute to the accumulation of amyloid beta, which in turn activates more microglia. The exact details by which neuroinflammation leads to the accumulation of amyloid beta, I do not know, as the paper did not go into the details. But over time, when these microglia become chronically activated, they become less effective at clearing those amyloid beta plaques. But they still continue to produce pro-inflammatory cytokines, which exacerbate neuronal damage and cause neural death. Microglia also interact with tau proteins, particularly in the later stages of Alzheimer's, which may contribute to abnormal tau phosphorylation and make it worse. Again, the paper did not go into details on this process, and I didn't have the time to investigate on my own. But it's important to note that there is a relationship between the two, and that neuroinflammation is intricately associated with amyloid beta and tau pathologies. Researchers have identified multiple activation stages of microglia, which show that their potential as a therapeutic agent in Alzheimer's is high because they're associated with different gene expression patterns. The paper also mentions that there are numerous drugs targeting inflammatory-related mechanisms under clinical trials right now, so we might see some of them as treatments in the near future. And I know that inflammation is kind of a big deal in a lot of other diseases as well, like cancer and autoimmune disorders, so I think it would be interesting to learn more about the exact mechanisms of how it affects Alzheimer's. Next, we have the metal ion hypothesis or framework. And this starts out by mentioning that trace metals, specific metals, maintain homeostasis of the neural metal microenvironment, a balance that is easily disrupted by inappropriate deposition or maldistribution of metal ions. And specifically, there are three that are closely associated with Alzheimer's disease, and they are iron, copper, and zinc in that order. You can see buildup of these metals in amyloid beta plaques and also neurofibrillary tangles. And that buildup plays a really critical role in the pathological protein deposition, which basically means that when biometals are in dyshomeostasis, so they're not in balance, they contribute to the buildup of harmful protein. In the case of zinc, for example, specifically, zinc can interfere with signaling through NMDA receptors, which we'll get to in a second, making zinc deficiency crucial in the context of glutamate excitotoxicity and dysfunction and Alzheimer's disease. We're going to talk about that hypothesis very soon, so we'll see what I mean. And when biometals are in dyshomeostasis, there's kind of two ways that they promote the aggregation or they mess things up. And one of them is the dysregulation of protein buildup, which means it leads to protein buildup, which interferes with organ function. And if those proteins cannot be broken down, then they end up causing neuronal death. The other effect is that metals are bound and stored within protein deposits, and they can initiate ROS production, reactive oxygen stress production. Again, we'll get to that in a second as well. But the essential thing here is that metal dyshomeostasis is closely linked to various events in Alzheimer's disease, such as amyloidosis, tauopathy, oxidative stress, and neuronal death. And research is fairly new in this framework and is still being conducted on how we can approach these issues. Reactive oxygen stress. We'll get to that in a second right here. So the paper kind of groups these two frameworks hypotheses together, but discusses them separately, so it takes a second to get to what it actually is. But the oxidative stress hypothesis talks about ROS, which is during normal metabolic processes, our body produces reactive oxygen species and other unstable, highly reactive substances. Reactive oxygen species are a type of free radical. I don't know if you've heard of them before, but they essentially break down everything they touch. They are unstable. They have unpaired electrons, free radicals, so they're really unstable and they want to bind to things. And so when they do, they change the structure of whatever they bind to, essentially destroying that thing. And ROS, reactive oxygen species, is one of those free radicals. And it's produced a lot by our metabolic process. Now, normally, these substances that are produced are neutralized by a really efficient antioxidant defense system that we have that protects cells from oxidative damage. However, in Alzheimer's, different factors such as metal accumulation, maybe overexpression of related enzymes, or mitochondrial dysfunction can stop our antioxidant defense system from dealing with these reactive substances and contribute to the production of excessive ROS, which results in a general imbalance. And that figure maybe is helpful. You see it's a normal stress free radicals, which are the group that ROS belong to, attack the cell, and then you have a cell with oxidative stress. And then maybe a good metaphor for this is kind of to think about an apple that is left out on the counter and it browns and ends up rotting over time. That's essentially what is happening to these cells when they're under oxidative stress. Oxidative stress presented in the early stages of Alzheimer's disease acts as a crosstalk between various hypotheses. For example, oxidative stress modulates the APP process, APP being the amyloid precursor protein that we talked about in the amyloid framework, and secretases activity, which promotes the amyloid pathway. For example, the activation of microglia induced by ROS triggers a neuroinflammatory cycle So you can see that these connections between oxidative stress and the other Alzheimer's frameworks have made antioxidants emerge as promising agents for Alzheimer's treatment. They have had some positive outcomes in animal models, but have yet to show efficacy in clinical trials. Several studies have shown that standalone antioxidant treatments haven't had significant cognitive benefits for patients with Alzheimer's disease. So the authors kept on recommending that more research needs to be done in the hypothesis, how it pairs well with other frameworks and things like that. Up next is...
Do we know why that happens? The oxidative stress. Do we know why that might occur in somebody and not somebody else?
That is, so it generally happens in everyone. Usually we have systems that have a really good antioxidant defense system that takes care of those and deals with those. But in patients with Alzheimer's, there are a lot of factors, some of which I've included here, that lead to just like a really excessive production of ROS, those reactive substances. So the defense system is overwhelmed and it can't deal with them anymore. One of those is, for example, the metal accumulation that we talked about in the previous framework. Overexpression of certain biometals, like maybe iron, zinc, and copper, can lead to excessive ROS. The specific, like exact details. Right. So yeah, the glutametric excitotoxicity hypothesis. So what is glutamate? Glutamate is the primary excitatory neurotransmitter in the brain and central nervous system. Glutamate activates NMDA receptors, the full name is right there, I don't want to pronounce it, that allows sodium and calcium to enter into neurons. Now, in normal resting membrane potential, there are magnesium blocks that prevent an excess of ion flow into neurons. Okay? But in Alzheimer's disease, chronic NMDA overactivation by excessive glutamate levels removes the magnesium block, which allows an excessive amount of sodium and calcium to flow into neurons, which you might be able to guess causes neuronal death. But excessive sodium specifically causes neuronal swelling, and elevated calcium levels initiate various processes, including the creation of more reactive oxygen species, more ROS than the system can handle, mitochondrial dysfunction, activation of necrotic and apoptotic pathways. And these processes ultimately result in permanent and cytotoxic damage to neurons, and they cause Well, I hope that at this point it's kind of starting to hopefully become clear how interconnected all these hypotheses are and how they all cause each other and are affected by each other, which is why developing treatments for Alzheimer's is so difficult. The authors mentioned that the effectiveness of neurotransmitter-based regulation in this context has been pharmaceutically validated, so they know that they have promising therapies in progress and that it works. But there are limitations, and there is a general focus on the other frameworks that has prevented research being done in this specific framework. They also mention that there's a potential for excitotoxicity to alter cognition levels earlier than amyloid and tau pathology. So it makes it a very important framework to focus on, and it might lead us to better therapies than the ones we currently have by dealing with those cognitive declines earlier than the other pathologies can show. So it holds great potential in Alzheimer's treatment. Ooh, this one is a good one. So the microbiota gut-brain axis. I don't know if you guys are on social media as much as I am, but in the past couple of years, there is this really big trend of everyone healing their gut microbiome. And there's so many influencers that are like, no, you need to drink so much kombucha and avoid all these foods to balance your microbiome. So I think a lot of research has been done in recent years that show how important the microbiome is in regulating our system and in dealing with other diseases. And of course, it's also a prevalent thing in Alzheimer's as well. I know Jordan has done quite a few presentations on this. I don't think he's online, but it would be cool if he had something to say about this. So getting into it, the microbiota refers to the collection of bacteria, fungi, viruses, archaea, and protozoa that live in the gut. These microorganisms offer nutritional and protective effects in metabolism and are also able to influence brain function via the microbiota gut-brain axis. However, certain factors such as maybe alteration in diet, use of antibiotics, prolonged exposure to psychosocial stress, irregularities in the immune system can shift the proportion of bacterial species in the microbiome, leading it into dysbiosis, potentially a state of unbalance. And when that happens, this change in the composition compromises the intestinal epithelial barrier, which can lead to harmful substances and penetrants entering in the intestinal tract to enter the bloodstream. This triggers an immune response that can lead to systemic inflammation, neuroinflammation hypothesis, and this systemic inflammation allows inflammatory mediators to cross over the blood-brain barrier and impact microglia in the brain specifically. This process is also accompanied by imbalanced neurotransmission, which ultimately leads to neuronal degeneration and death. Here you have kind of a little graphic of the gut-brain axis. It was frustrating me because when I think of axis, for some reason I think of straight up and down and not a circle, and so all of the things were showing a circle. But the paper mentions that the investigation of this mechanism is still at an early stage because it's pretty recent development that they found, and that the exact ways in which the gut microbiome affects brain activity or its connections with the other pathologies of Alzheimer's are still unknown and are the focus of current research. And then we have one more, the abnormal autophagy hypothesis. Autophagy is a metabolic degradation process that maintains cellular homeostasis by delivering damaged or old cellular components to lysosomes for degradation and recycling. I have a little chart on the right there, which hopefully explains it pretty well. Autophagosomes are just saying kind of like sacs that carry broken down or damaged parts of cells. And they form and they travel along microtubules to the microtubule organizing center, which has a lot of lysosomes. The lysosomes fuse with these autophagosomes to form autolysosomes. And the autolysosomes are where organelles and other cellular malnourished or stressed neurons. There is a normal accumulation of autolysosomes, and that has been linked with amyloid beta buildup, which suggests that defective autophagy is closely linked to amyloid pathology. This is another one where the paper did not go into much detail and said that it's still developing, but in general, the abnormal death of neurons is also its own hypothesis. There is increasing evidence that links or indicates that genetic factors, reduced expression of related proteins, and defective transportation are potential causes of autophagy disruptions, but still more research is being done in this framework. So yeah, those are the frameworks that the paper discusses and encourages researchers to work within those or with a combination of those to develop more therapies for Alzheimer's. If there are any questions, I'd be happy to answer.
So everything answers.
Everything, yeah, that was really
And
good.
yet not all people have it.
Yeah.
How's that possible?
It's, I don't know if it was helpful or not, but I tried to kind of present this at a very high overview, but when you go into the details, it's very complicated. Everything is so interconnected with each other, and there's no, even in these hypotheses, they're all like, well, this may lead to this, and may lead to this, and we still don't know how all of these interactions work. And it becomes, I don't know how they do it, because like they don't know anything for facts, the way that Alzheimer's develops, or the specific details of it. At least that's the impression I got from this paper. So
And so looking at this particular six stories for how you're going to be looking at imaging going forward, like do you feel stronger in the idea that it's a functional change versus Alzheimer's, or you're skeptical of that, or how is it affecting something imaging?
So that's actually one of the reasons I picked this paper to begin with. I did not know that much about kind of the background of how Alzheimer's works, and I wanted to eventually work on some projects related to it. And I think it's really helpful to approach those projects kind of from the understanding of one of these mechanisms. I still don't have a made up opinion about how I think it impacts certain things because I've never worked with Alzheimer's data specifically. But I did this for myself so that when I do get there, I have an understanding of how to approach the problem.
Yeah, just thinking about maybe one of are there any of these that jump out at you as potentially like having large effects on how the image or what we see in imaging, or just
I mean, the plaques are kind of the big one, but that's kind of what everyone already generally focuses on because they're so easy to see. Out of all of them, I'm particularly interested in more of this one, the microbiota gut-brain axis, and how we can potentially see this effect or manifest itself in data, neuroimaging data, functional data. I think this one would be
And I mean, I had a question about your slide to the amyloid hypothesis. I think you actually had some images of brain areas that were affected. Yeah, are these like functional regions or just actually structural brain regions?
okay. I just found the picture of projects because it was really difficult finding figures that showed any of these hypotheses because they were so technically detailed. I have linked the paper down there, so I can look and let you know whether it was functional.
like this is just a box of slides comparison structural.
And so I kind of like getting into what we're asking, like what causes the dyshomeostasis in the metal compounds?
The metal compounds. This was one of the ones where the paper didn't go into it and still said that it's a large area of research. I know they did mention that possible reasons. They did. I don't.
Yeah, maybe there's some like process that gets confirmed because of Alzheimer's.
Maybe I assume it's partially in combination with the other mechanisms. When generally anything goes out of balance, it leads to other things going out of balance. So I think it starts like a domino effect thing. I don't know, to be honest.
You talk about the kind of case study to questions about so I was wondering if you, like in this paper, or maybe people conducted different stories about how symptoms progress, or things that maybe things that can answer.
I read it.
I guess what's like that you're interested in this thing.
I just had a general interest in AD. My grandpa had Alzheimer's, so I was just kind of like, what is even happening? So that's where I decided to look into this. And I know a lot of people work with Alzheimer's around us, so I was like, if I ever end up going into that direction, it's for me. I don't think this paper is necessarily particularly helpful for kind of setting up an area of or an approach in research for our field, or specifically with neuroimaging. I have to look more into kind of how we can combine or maybe reinterpret or come up with other hypotheses that can be used for the data that we have. But I haven't really.
It's a different topic. Like, which treatment, like, so you talked a little bit about treatments, and then there are like some that differ based on the underlying mechanism.
So the paper itself, let me give them the credit that they deserve. They talk about a lot of other stuff. In addition to this, they have like a whole 10 pages on the different signaling pathways in the brain and in the body that are also maybe potential ways of looking at Alzheimer's disease progression. They talk a lot about the clinical trials that are currently happening for all of these hypotheses and the specific. So they have all like the specific. I don't know what it's called, but the structures of diseases or everything that they use. So they go into a lot of detail about this, but it's just so much technical detail that one, I don't understand it, and two, I don't know how to relate it in a way that would be helpful for anyone.
Well, if you looked over the whole paper, they talk about treatments that cut across the different mechanisms, right?
So no, they haven't specifically talked about one that's crossed. They do talk about combining therapies that target each of them separately. So for example, they would combine an antioxidant treatment for the neuroinflammation with one for amyloid beta. And that's what they kind of mention is the general approach of how therapies are going currently. And there would need to be more work to lead into one that maybe addresses multiple.
No, you mentioned that it's gonna lead to something that is
It might. I mean, research is so.
like you just put it there.
Yeah.
Okay.
Yeah, but looking at this, at least 8 or 9 classes of possible ways for our organism to fail only to one disorder.
Yeah.
I wonder, it's just amazing how this all works. We never build anything that even remotely is like that. It's anything like we're not like why not anything on is rising. Although no changes are made, no thread. It stops working just by itself. And the argument that I constantly fighting. How does anything even work?
Very good question.
So, thank you for doing this. I understood maybe 50% of words that you said. Yeah, thanks everyone for joining. Any more questions? No. Maybe from people online. I don't know. I didn't see any comments there. No. Yes. So.